Alright folks, since I keep forgetting to do this in lab, I thought I would run through some things that you are going to need to know for the first lab quiz. And so really what I'm going to do is I'm going to talk about, or I'm just going to work through the different labs that we've done in class, and that should remind me of the number of things that I usually ask. So a little bit about the format of the quiz. I'm probably going to put it in Socrative and have you take it that way. It'll be a collection of pictures that you need to identify, results of stains, for instance, so gram stains or endospore stains, just general simple stain. So you'll have pictures like that. You also have maybe a few multiple choice things, and then a few fill in the blank things or short answer things as well. So lab one was microscope. So anything there. It could have been how to calculate maybe a total magnification. That could be something that I ask. It could be about the oil immersion lens and why we use that, not the oil immersions. Why is it that we use the oil itself? That could be a question on that. When we typically use oil on the 1,000 times magnification lens. So why is it that we use it there and nowhere else? When is it that you use the coarse versus the fine adjustment knob with the scope? Remember, the only times you should use coarse adjustment is on the very lowest power magnification. No other time should you use it there. If you're curious about the oil immersion, remember that if the oil lens looks something like this, right, and your slide is down here, as you increase magnification, you decrease this working distance. And so as you decrease that working distance, you also decrease the aperture, right? So it might become that small now. And so light has a hard time getting into that tiny little hole. So as light comes through, it'll hit the lens and then it will bend. And so the reason that you want the oil there is so that it'll create a boundary, and that boundary layer, the oil will bend light just like the slide will, so that now what's going to happen is the light will come up through and it'll bend a little bit, but it'll still get captured by the aperture in the lens. So again, if this is the 1000x lens, right? And so that's the purpose of the oil. All right, what else on microscopes? Total magnification, lenses. I think that might be it. It's a pretty simple lab. The second lab that we did, I just had you look at cell types. If I recall, I think I had you look at some cheek cells maybe. My pen's crapping out on me. Actually, I think I called them lip cells for you guys. I had you look at some yogurt. I had you look at just a variety of different cells, and really the point there was to see the difference between the size of prokaryotic and eukaryotic cells. I'm not going to give you pictures of cells and have you identify them necessarily, but I would expect that you would know general differences as far as size there. In one of the next labs, we talked about, let's see, we talked about aseptic technique. So what is aseptic technique? That's certainly something that I would like you to know. Why do we use it? Remember, there's 3 reasons why we use aseptic technique. So you should know those. Remember, that's to protect you, protect your sample, and protect the environment. Why is it that we flame the lids or the loops when we're doing aseptic technique? What's the purpose there? What's the proper way to store plates? Which, judging by the last lab, a lot of you still haven't figured that out yet, so think about that some. And then here's where I would probably ask you a question like, how is it that you transfer media from, or a bacterium from one thing to another? So from a slant to a broth, or from a broth to a plate, or whatever. I might would ask you up here in the microscope stuff, a short answer could be just to kind of walk me through the process of using the oil and the oil lens, and then how do you correctly clean up from that. So that's another possible short answer question. We did gram stains. Right, so again, what do those show us? How do you do them? The gram stain will be the only stain that I want you to know all of the stains and liquids that we used. So you started with the crystal violet, then you washed with water, then the iodine, washed with water, destained or decolorized with alcohol, rinsed with water, and then saffronin. You don't have to know times for those, but you need to know the orders of each one. We did smear preps. So again, why did we do those? Why do we do smear preps? When do we do smear preps? That's all I can think there. With the gram stains, you would also need to be able to ID them. So if I give you a picture of a gram stain, can you tell me whether it's positive or negative, for instance? We did endospore stains. So here again, I could ask you to ID them. So if I give you a picture of an endospore stain, can you tell me whether it's positive or negative or not? Can you tell me when bacteria produce endospores? So if I had a 24-hour culture and you did not find endospores on there, why might that be or why not? Think about that. What about if I had a week-old culture? Would you expect to find endospores there or not? So those are some questions that I could probably ask. Why is it, so maybe also with the endospore, why is it that we had to use heat to do the endospore stain? Remember, that's the one where we put the slides over top of the steam, and that's the only way it would work. So why is it that we had to do that for endospore stains? Right, we did simple stains. Right, so I'm kind of doing this a little bit backwards. So we did the simple stain. Right. What does a simple stain not tell us, for instance, that maybe the gram stain does? And if I do the simple stain itself, what does it tell us? Remember, about the only thing you get from the simple stain is, are they cocci bacteria or are they bacillus? Right, and those are the correct names for those. And you can see them, but other than that, the simple stain doesn't tell you a whole lot. It's just kind of a quick and dirty, do I have bacteria there available? We did a pH and temperature lab. And so that was just looking at the effects of pH and temperature. I know we didn't do the stats with those, but I think you got a pretty good idea of kind of how temperature and pH can affect bacterial growth. The thing I may ask here is how did we measure this? So how is it that we looked at the effects of pH and temperature on growth? What did we use? And how does that work? Right? So we used the spectrophotometer. the spec, it's called the spec 20 or the spectrophotometer. And so remember, that just shines light in and measures the amount of light out, and then you should be able to interpret how much growth you get based on that. Let's see, what else did we do in the pH? I think that was about it. Then we did the endonuclease lab. Well, that was the next one I asked you a bunch of questions on anyway. So do you know what endonucleases are? Are you able to interpret a gel if I give you one? For instance, maybe even for paternity, right? So if I give you a gel that looks like this, whoops, crapped out on me there. And so maybe what I have is a banding pattern that looks like this for one individual, this for another individual, and then I want to know if this last individual is related or not, and so I could do something like that, and I would say, who is this third individual related to? Can you interpret that? Or if I give you one like the one that I actually posted in lab, an actual screenshot of yours, and asked you to troubleshoot it based on what's there, could you do that? So those were the endonuclease things. The other thing that we did that I didn't ask you questions on had to do with, remember we did isolation streaks, right? So remember you had your plate, you struck here, you flamed your loop, you drug, you struck through, you flamed your loop, you drug, you struck, you drug, you struck. And so how do you know how would you have known if you were successful there? Well, remember, the only thing you're looking for is if you have nice individual colonies somewhere. So it doesn't have to be, so maybe the first quadrant looks like this, right? And the second quadrant might look like this. And then by the time you get to the third quadrant, you actually start to get individual colonies, right? And so that's all you're looking for. If you see individual colonies like that, then that means you were successful. So let me pull one up here and show you a picture. Let's see. Like here. So that's a successful isolation. So this was probably their first zone, then their second, and then their third. They may have only done 3 here, because once you get good at knowing how to manipulate bacteria, then 4 is kind of overkill. But as long as you see these individual colonies, then you were successful in isolating bacteria, and so that's what you're looking for. And again, I'll be pretty straightforward with what I'm after. As far as gram stains, right? So let me do this so you don't get a mixed culture. Whoops, not gram T. So if I gave you one of these pictures, could you tell me whether they're gram positive or gram negative? Now, obviously, these are already labeled for you, and so you wouldn't get that in my quiz. But can you tell me one way or the other there? Right. Oh, I could also do endospore. If I could spell it correctly. So let's say you saw this one maybe. Right, so is that positive or negative for endospores, and how do you know? That could be something else that I would show you picture-wise. I could just show you a picture. Maybe this was a simple stain instead of a gram stain. I might would ask you to tell me what are the shape of the bacteria. So obviously I would give you one that looks a little better than this, but that is a question that I might ask. And so let me see if there's anything else. Oh, so we just finished in lab, we just finished the transformation. Lab. So even though you won't be able to see your results yet, what I'm after there is just general stuff. So what are you looking for? What was the transformation? How would you know if you had successfully transformed the bacteria or not? What is it we're looking for? And those should all be questions that you should be able to answer without actually seeing your results, and whether or not these actually end up working, that's another issue. And so I think that's pretty much what we covered. If I think of anything else, I'll try to mention it. If you have any questions, let me know. Just wanted to create this and let you guys know. We will have this first quiz. Let's see, today is the 3rd. So we'll have this first quiz on the 8th, and then we'll have the last one at the end of the semester, which will cover everything else that we do in lab. So again, if you have any questions, let me know.